Welcome to Pramod YouTube channel. Now I am going to discuss the mechanism of action of anti-cancer agents. Anti-cancer agents. Anti-cancer agents. For my previous video, if you go through in my previous video, I have given you the mechanism of action of first class of anti-cancer agent, that is alkylating agent. Now I am going to give, you can follow that link and see regarding the first class of anti-cancer agent. Now I am going to deal the mechanism of action of second class, that is anti-metabolites. Anti-metabolites. So what are these anti-metabolites? Anti means against the metabolites. What are these metabolites? The compounds which are formed during the metabolism are called as metabolites. Now what happens? These are structural resemblance to the metabolites. So now I would like to tell what is the mechanism of action of anti-metabolites. Now anti-metabolites, they are structural resemblance to metabolites. How they are structural resemblance to metabolite, I will explain you. And they are capable of binding the metabolites, thereby inhibiting the DNA synthesis. How they inhibit the DNA synthesis, I will explain you. What actually the metabolism takes place, how the pathway takes place, I will explain you regarding in detail. Now antimetabolites classes are folate antagonist, folic acid antagonist, purine antagonist, pyrimidine antagonist. So that you already know, we have discussed in my previous video, I have discussed it. Now these antimetabolites, how they act as an antimetabolites, I will tell you the pathway. Para-aminobenzoic acid combined with pteridine, it forms dihydroteroic acid. Dihydropteridine synthetase. Dihydropteridine synthetase. With the help of this, dihydroteroic acid is formed. This combined with glutamine forms dihydrofolic acid. Dihydrofolic acid forms tetrahydrofolic acid. Tetrahydrofolic acid is responsible for purine and pyrimidine synthesis. Of DNA. This helps in the DNA synthesis. So this is the pathway how the DNA synthesis takes place. Now dihydrofolic acid is converted into tetrahydrofolic acid by enzyme dihydrofolate reductase enzyme. Now this is in a general pathway where the DNA synthesis takes place. Para-aminobenzoic acid combined with the pteridine. In my previous lecture, I told you that pteridine means pyrimidine fused with pyrazine. Both combined together by dihydropteridine synthetase enzyme in presence of it. It forms a dihydroteroic acid. This combined with amino acid glutamine, it forms a dihydrofolic acid. Dihydrofolic acid will convert into tetrahydrofolic acid by dihydrofolate reductase enzyme. This tetrahydrofolic acid, by many steps, it synthesizes purine and pyrimidine, followed by responsible for DNA synthesis. This is the overall general pathway of DNA synthesis of normal cells and cancerous cells. Now what happens means, our particular drug, what they do means, this antimetabolites like methotrexate. So methotrexate, what it will do means, it will inhibit the very important step, folate antagonist. Why we tell folate antagonist means they are structural resemblance to dihydrofolic acid. I told you antimetabolites means like methotrexate. Methotrexate folate antagonist. It will bind and inhibit the dihydrofolate reductase. As a result, there is no conversion of dihydrofolic acid into tetrahydrofolic acid. When dihydrofolic acid is not converted into tetrahydrofolic acid, there is no DNA synthesis, there is no cell division takes place, there is no proliferation of cancerous cells takes place. So methotrexate is a class where they bind or inhibit directly on the dihydrofolate reductase because they are structural resemblance. Methotrexate is an antimetabolite. This is a metabolite. So I told the metabolite, antimetabolites are structural resemblance to metabolites. So dihydrofolic acid and methotrexate contains, methotrexate also contains pyridine. It also contains para-aminobenzoic acid. I have given you the structure. You go through with the structure. So similar structure of dihydrofolic acid. So instead of dihydrofolic acid converting into tetrahydrofolic acid, methotrexate bind to dihydrofolic acid reductase and inhibit. Thereby there is no conversion, no DNA synthesis. One way. The other way, the purine antagonist. Purine antagonist example is 6-mercaptopurine. 6-mercaptopurine. So the purine which is responsible for DNA synthesis are inhibited by purine antagonist. They are also their structure is also structural resemblance to purine. So as a result, there is no DNA synthesis. So they will inhibit the purine directly. Pyrimidine 5-fluorouracil. 5-fluorouracil. This will bind directly with the pyrimidine metabolite which is formed, pyrimidine, and there is no DNA synthesis. No DNA synthesis. As a result, what happens? There is no proliferation of cells. There is no development of cancerous cells. This is all about the mechanism of antimetabolites. Now let us move on to the third mechanism of action is antibiotics. So antibiotics is also very important. How does this antibiotics will act? Third one is antibiotics. Antibiotics example is donorubicin, doxorubicin, bleomycin, dactinomycin, all comes under the class of anti-cancer antibiotics. Now this anti-cancer antibiotics, what they do means, when they enter into the body, when the antibiotics are given, they form a superoxide. And hydroxyl free radicals. Free radicals are very dangerous. This superoxide, the antibiotics like donorubicin, doxorubicin, they generate, what they do, they generate the superoxide or hydroxyl free radicals. Hydroxyl free radicals are very toxic to the cancerous cells. Okay, so what these free radicals or superoxide hydroxyl do means, they will be inserted in the DNA structure. They will be inserted in the DNA structure. They will be inserted in the helical type of DNA structure. Okay, so this superoxide of the antibiotic, what they do means, they insert on a DNA structure. Say that I will mention in a green form. So superoxide free radical insert into a DNA. Super hydroxyl free radicals of donorubicin, doxorubicin insert into a DNA structure. When it insert into a DNA structure, disruption of DNA takes place. What is going to happen? disruption of DNA, disruption of DNA, disruption of DNA. As a result, there is no proliferation of cancerous cells, no proliferation of cancer cells. So they directly insert. How they insert means by generating a strong free radicals, reactive free radicals, which insert into a DNA, disturb the structure of DNA. Thereby, there is no further proliferation of cancerous cells. This is how the antibiotics actually act. So this is in a third class of anti-cancer agent. Now, last important class of anti-cancer agent is in a natural drug, Vinca alkaloids. Okay, so how does this Vinca alkaloids act? So fourth one is Vinca alkaloids, Vinca alkaloids, Vinca alkaloids. So how does this Vinca alkaloids act? So Vinca alkaloids like the drugs like vincristine, or vinblastine. This will directly arrest the metaphase of the mitosis of cell division. Where they will directly attack means they attack on the metaphase of the mitosis process. They bind with the microtubule. How they bind with the microtubule? I will give you with the schematic representation. Now we take that microtubule, say the microtubule protein during the mitosis, during the cell division process. This is very important thing. Okay, so here if you see that this all are very important. Now this microtubule, say this is microtubule. Okay, so microtubule, what they do means reversibly they form a dimers. Dimers means 2 they get separated. One is this, one is this. Okay, let me draw first. They form a dimers. Okay, they form a dimers. So dimers will be formed. Now when dimers are formed, in general mechanism what happens? Dimers will convert into polymerization. They will undergo polymerization followed by the cell division. Polymerization, many are formed, n number are formed. Dimers get separated, then they form a many number of the polymerization takes place, thereby cell division. So there is a cell, dimers is converted into polymers happen. Now what happens? This is a normal cell division. So microtubule or during the mitosis of the metaphase, they convert into dimers. They get separated and convert into dimers. They will undergo polymerization. This is a normal mechanism. Now what does the drug will do means, you take the drug, for example vincristine or vinblastine, which is having a indole as a nucleus, basic nucleus, indole. So what this drug will do means, I represent like this, the drug structure. This structure stop the polymerization. You say this is a vincristine or vinblastine. Okay, so this is a drug. Now what happens? That drug will cover with the dimers. So what happens means, I draw first black. Bind with the microtubule. They are binding with the microtubule during the metaphase and arrest the metaphase. Now what is going to happen means this is going to cover. Yes, the vincristine or vinblastine act on the dimers and they form this bind with the dimers and they prevent the polymerization, thereby cell division. So vincristine vinblastine binds with the microtubules and prevent the polymerization. By binding with it, it prevents the polymerization. There is no proliferation of cells, there is no cancerous cells, hence the death of the cancerous cells or proliferation of cancerous cells or the proliferation of the cells will be stopped. So vincristine and vinblastine act by inhibiting the mitosis by binding with the microtubules, that is the binding to the dimers and preventing further polymerization. This is what the mechanism of overall important class of anti-cancer agents. I have not gone to all class but important class of anti-cancer agent. Thank you.